My lab is fundamentally interested in what metabolic activity occurs in biological systems, including cancer. How can we measure it, and how can we control it? And to make progress from that basic question that my lab focuses on to the broader cancer problem, we really need collaborators who are masters of cancer biology, masters of the latest computational tools to take advantage of the very complex data we generate, masters of the immune system. And that's what I get from being part of the Ludwig Princeton branch. You know, we have some sense from where in the world people live long lives of what's generally a healthy diet. And we know something about what kind of diets prevent heart disease, and that data has been really impactful. We've seen heart disease rates really go down. We just don't have the equivalent knowledge for cancer. And it's such a distinctive situation that a cancer patient finds them in. Their body is going through such distinct biological events that the common dietary wisdom may be totally wrong. It may not be simply eating healthy in the classic way. It may be really zoning in on specific nutrients that are needed for the immune system to fight tumors, or specific dietary makeups that suppress the signals that drive cancer. And that's really what we're looking to uncover. There are three directions that we're currently pursuing that I'm particularly excited about. One is trying to get more definitive answers on whether ketogenic diets actually work in cancer, and if so, how. We're expecting results later this year on a clinical trial where patients are randomized to get either a normal diet or a ketogenic diet combined with the best possible chemotherapy to treat metastatic pancreatic cancer. And whatever the outcome is, it's going to tell us a lot about the directions to pursue in terms of how to metabolically target cancer. Another direction that we're pursuing is trying to supplement what's missing in immune cells when they're fighting the tumor. And specifically, we're looking at something that's historically been considered a poison, methanol, as a way to give to these tumors tiny, tiny carbon units that the immune cells in the tumor particularly need in order to build themselves up so that they can kill the cancer cells. It's an example of the really high-risk work that's very hard to pursue otherwise that's enabled by Ludwig, and the results in mice are really dramatic. A third direction we're currently pursuing is what are the metabolic triggers that would kill cancer cells, and particularly get them to kind of commit suicide or die by apoptosis. Right in the chemistry building at Princeton, a drug was made called pemetrexed that's first-line treatment for lung cancer by shutting down folate metabolism. And so we're learning about how pemetrexed does that to cancer cells. We're really interested in why it doesn't do the same thing to immune cells. How are they exempt? How can they keep fighting the tumor while the cancer cells are being poisoned? And what can we add on top of this to make those cancer cells die faster? I think a really powerful part of Ludwig is that we have the resources to do the very best work on our areas of expertise within our labs, and then we also have other resources to go beyond what we can do in our labs and to do the most impactful possible synergistic projects, focusing solely on what will make scientific breakthroughs that help patients without any constraints of lab boundaries or funding.